In the last lecture, we looked at the many different organelles that must function in order to keep a cell alive. In this lecture, we're going to take a look specifically at the organelle called the plasma membrane. We're going to take a look at its structure and its many different functions. Cell membranes, including the plasma membrane and also those of like the endoplasmic reticulum and the Golgi apparatus, are going to consist of lipids and proteins. So they're going to consist of fats and proteins. And it's a special category of lipids called phospholipids, okay, that are in cell membranes. And in phospholipids, we have two fatty acid chains that are attached to a phosphate group. And so we're going to take a look at that structure of the phospholipid because it's unique to cell membranes and it's very important for their function. Just like we talked about nucleic acids and their phosphate groups, these phospholipids phosphate groups are electrically charged. They have extra electrons. They're going to give it a negative charge. And because they're charged, that means that they are going to be attracted to water. So the phosphate group in the phospholipids, which make up the cell membrane, are what we call hydrophilic because they like water. Okay. Now, remember we talked about the fatty acids being hydrocarbons. They're nonpolar, and so they are hydrophobic. So they do not like water. They are water hating. They want to avoid water. So this sets up kind of a dichotomy in the cell membrane where we have a water-loving part of it and a water-hating part in the middle. This is what we call the phospholipid bilayer of membranes. So we have two groups of these phospholipids, and their hydrophobic tails are kind of in the center of the membrane, and the hydrophilic heads are on the outside. See, the outside of the cell, or either kind of the inside of the cell touching the cytoplasm. So this slide just goes kind of a little deeper about what I had just explained. The phosphate group is going to like water. It's going to mix well with water, whereas the fatty acids are going to hide from water because they don't like it. So again, that it gives a two-layered membrane look to this phospholipid bilayer, with the hydrophilic heads being in the middle that don't like water, and the hydrophobic parts in the middle, and the hydrophilic parts on the outside. So they can be attracted to water. Cytoplasm is made mostly of water, and usually the outside of the cells are going to be interacting with water-based molecules. Now, along with the phospholipid bilayer in membranes, there's also many proteins found amongst the phospholipid bilayer. And they're specific proteins for specific jobs. And the interesting part of them is that they're not fixed in the membrane. They actually can move around in the plane of the phospholipid bilayer. So it gives the membrane kind of a fluid nature, where the proteins can actually kind of move around back and forth within it. So we call that the fluid mosaic model of a membrane, because nothing is rigid, kind of set in stone. It's very fluid, and lots of movement can happen within the membrane. So there are many different types of membrane proteins that exist with many different kinds of functions. One type of protein is going to attach itself to the cytoskeleton on the inside of the cell, and extracellular matrix fibers on the outside of the cell, so that the cell will maintain its shape and kind of stay fixed in place wherever it is in the body. Other proteins in the membrane serve for cell signaling. So they bind chemicals from the outside and are attached to signaling proteins on the inside that kind of relay that message from the outside to the inside, telling the cell what to do. We can see examples in the picture of these two types of proteins. The cell shape maintenance protein, would look like this, where it's attached to the the extracellular matrix on the outside and the cytoskeleton on the inside. And the cell signaling looks like this. So if this was a little cell signal, that triangle piece on the outside, that's like a hormone or something that is going to send a message to a cell. It will come and attach itself to the protein in the plasma membrane, and that then sends the signals inside of the cell to tell the cell what to do. Some membrane proteins are performing chemical reactions, so they're enzyme proteins, just like these ones right here. So these proteins are in the cell membrane, but they're all kind of tucked in there doing some chemical reactions for the cell. Other ones are transport proteins, which are like little tunnels between the outside and the inside of the cell, like this example right here. So they form tunnels that can let substances into the cell or out of the cell. Other membrane proteins are for cell-to-cell recognition, so that the cells can recognize each other and make sure that one of them's not an enemy or an invader, a bacterial cell or a virus. So that is shown right here, cell-to-cell recognition. And then finally, we also have proteins in the cell membrane that are for intercellular joining. So they're going to actually hook up with the proteins in another cell membrane, and they're going to lock themselves together so that those two cells are joined together to make a tissue. When we take a look at animal cells, we will remember that they don't have cell walls like plant cells, but animal cells do have what's called an extracellular matrix kind of on the outside, and we alluded to that when we talked about those proteins that hook onto the extracellular matrix to provide support for the cell. So that extracellular matrix, again, is for support, but can also be kind of an added extra layer of protection for the cell. Now, between cells, we can have three types of junctions, okay, or joining together of cells. The first type is what we call a tight junction, okay, and that's basically going to bind two cells together so that they're anchored together. They can't, they're not going to drift apart and move around. And that's important for keeping tissues together so that they function for whatever purpose they're for. And these tight junctions are so tight that no substances can move between the cells at that point where that tight junction is. So it's like, it basically is holding those cells tightly together so that they don't move away from each other. The second type of junction is what we call anchoring junctions. Okay, and that's shown here and here. So these, again, they serve to hold those cells together as well, but they're a little looser than tight junctions, and they can allow some substances to pass between the cells, whether it's signaling substances or water or any other important proteins and such. Now the third type is a communicating junction, and that's basically kind of like a tunnel between two cells. So water and ions and proteins and stuff can pass from cell to cell, so they can kind of share each other's resources in a sense. So the communication junctions are kind of like this. So they're little tunnels between cells that they can pass substances through. These communicating junctions also allow cells to become one in function as well. So when we've got a whole bunch of cells together to make a tissue and a tissue to make an organ, they need to communicate with each other in order to stay in kind of synchronized fashion in order to carry out their function. All right, so let's take a look at plant cells. Like we said earlier, plant cells have cell walls that help them to protect them, maintain the shape and stuff like that, so they don't really need any proteins in their cell membrane to kind of hold them there like we saw in the animal cells. Cell walls also prevent excess water intake so that they don't burst. But plant cells, just like animal cells have to communicate with each other and they have to coordinate their functions, so they do have little junctions between them here, which we call plasmodesmata, okay, which are basically channels between the cells, so that nutrients and water and all that stuff can be shared. Signaling proteins can be carried through these so that they coordinate their function and they're all doing what they're supposed to be doing. Alright, so for a cell to survive, it needs to be able to take water, nutrients, other things inside, and also get rid of waste and send out signals and stuff of its own from inside the cell membrane outside to other cells. So we're going to talk about membrane transport. So the primary function of a plasma membrane is to regulate the movement of materials in and out of the cell. So that's the main function of a membrane, is to regulate what goes in and what comes out. And those transport proteins that we had talked about earlier, the ones that are channels, are going to help with this transport function. If we're talking about membrane transport, we need to talk about the types of transport that can occur. And one type of transport is called passive transport, or another word that you probably are familiar with is diffusion. And in diffusion, molecules are going to go from high concentration, through a membrane to low concentration. So it's kind of the same if you open up a perfume bottle, there's a ton of perfume molecules inside the bottle, and they're going to slowly make their way out into the air where there's not many perfume molecules. So we can see that here in this picture. We've got, we start out with a high amount of molecules on the left, and as soon as we kind of let those diffuse through, they're going to go through the membrane until it's equal. Okay, so until we've reached equilibrium. So there's equal numbers of molecules on both sides. Now this is what we call diffusion. The same thing can happen if we have two different types of molecules. Say if we're separating them from each other at first. Okay, so we've got a high concentration of blue on the left and a high concentration of red on the right. Well, if we open up the gates between them, again, they're going to diffuse and they're going to go from high concentration to low concentration again until we get an equal number of molecules on each side. So that's kind of the basis of diffusion. So similarly to the common diffusion examples we know of, freshly baked cookie aroma, perfume aroma coming through the air, plasma membranes also can facilitate diffusion. Okay, so diffusion, depending on what type of substances can diffuse, can go through the plasma membrane, but the plasma membrane obviously doesn't let everything in through diffusion. So it acts kind of as a barrier. So we call the plasma membrane a semi-permeable membrane because it allows some things to come through, but not everything to come through, right? So small molecules, lipid-soluble molecules can move through the plasma membrane pretty easily in what we call diffusion. Or passive transport, but others we're going to need a little more help. So this distinction between which molecules can come through easily and diffuse through the membrane versus which ones can't is basically what we call the cell's selective permeability. Okay, so it will be selective on what it allows to diffuse through and what it does not. Remember, diffusion always goes, like I had said before, from high concentration to low concentration. So if you have a lot of salt water on the outside of a cell versus the inside of the cell, that salt is going to diffuse into the cell, not out of the cell. Okay, it's because it always goes from high concentration to low concentration. All right, diffusion, or what we call passive transport, is called passive because the cell doesn't need to use any energy in order to move those molecules. Okay, it just happens through the nature of diffusion going from high concentration to low concentration. There's no energy that the cell needs to expel in order to move these across the membrane. So again, like we said, small molecules can usually get through the membrane without any help, and they're good. However, if they're bigger molecules, okay, or molecules of certain charges and stuff like that, we're going to need a little bit of help. We're going to need to exert some energy in order to get these particular substances inside the cell. So that's what we call on the transport proteins, those proteins in the membrane, okay, which need to use energy to open up or close in order to facilitate the diffusion of some molecules. So when water diffuses across the plasma membrane, we call that osmosis. So we're going to take a look at 3 different types of solutions depending on the concentration of water and solutes, on one side of a membrane versus the other. In this picture, we have 2 solutions that are divided by a semi-permeable membrane that is represented by those dashed lines there. Now, if we see in the picture on the left, we have a solution with greater concentration of solutes on the right-hand side, and that's what we call a hypertonic solution. So basically, another way we can think of it is it has more sugar molecules than water. Okay, so it's got a lot of sugar molecules and kind of less water molecules than the solution on the left, which has a lot more water molecules and a lot less sugar molecules than the other solutions. So we call that hypotonic. Okay, so if the membrane there is semi-permeable and it's not letting the sugar molecules get through, then it will let the water molecules get through. And so what happens is that we get osmosis, or movement of water, in order to kind of balance out the tonicity of the solutions. So the water will go from greater concentration, which remember we have on the hypotonic side, that means we've got more water than the other, to, so it's going to go, water is going to go from left to right into the hypertonic solution so that it kind of dilutes it out a little bit, and in the end we get an equilibrium or an isotonic solution. It's got equal numbers of the same ratio, is probably a better way to say it, the same ratio of sugar molecules and water on both sides of that membrane. But remember that the sugar molecules could not move because of that selectively permeable membrane. Only the water molecules could move. Water balancing is one of the most important functions that a cell membrane can do, because it will basically determine on whether a cell is nice and turgid, or whether it bursts, or whether it shrivels, depending on the movement of water. Because remember, water is one of those molecules that diffuses easily through the cell membrane. So cells are usually, thankfully, around isotonic surroundings. So they've got equal concentrations outside the cell as inside the cell. So water can move in and out equally. So there's not really a net movement of water. It's going to be equal. Now, if we put that cell, though, in a hypotonic solution, okay, remember, hypotonic means that there are low solutes, right, and lots of water. So basically, we're putting it in an environment where there's a lot more water molecules on the outside than the inside. Well, what happens? The water goes from high concentration to low concentration. It's going to go, the water is going to move inside, okay, that cell faster than it can move out, and what happens is that this, an animal cell will lyse, and the plant cells will start to fill up and get too turgid. Now, on the other hand, if we have a cell in a hypertonic solution, okay, remember, hypertonic means lots of solutes, right? Low amount of water. Okay, so we've got a lot more solutes on the outside, low water on the outside. So what happens? The water molecules are going to go from, remember, high concentration to low concentration. There's more water molecules on the inside of the cell than the outside, then the water is going to escape and go out of the cell, shriveling it. Okay, so water balance is super important to cells. Too much water on the inside can burst them. Too little water on the inside can shrivel them up. It is interesting to note that animal cells and plant cells require different tonicities of solutes in order to keep them healthy. So we said for animal cells that an isotonic solution is best. If it's hypertonic, then the cell will shrivel. If it's hypotonic, then the cell will burst. However, in plant cells, they have that rigid cell wall. And so a plant cell in an isotonic solution is actually going to be kind of wilty or floppy. Okay. If it's in a hypotonic solution, where they have a net water inflow, it's actually going to keep the cell real turgid and stiff and healthy in a sense, because that plant cell wall is going to kind of hold it in place, and it's not going to be too flimsy, and it won't really allow it to burst. And now in a hypertonic solution, where there's more solutes on the outside of the cell, that plant cell is going to lose water to the outside, so it's going to get all shriveled up and it's going to die. It's going to go through plasmolysis and die. So interestingly, that again, animal cells really require an isotonic solution in order to stay healthy, but plant cells require a bit of a hypotonic solution to stay healthy. Okay, so diffusion and transport, those are passive transport types of movement where, remember, there's no energy required. The molecules, the water molecules, are the very small molecules that can go through the plasma membrane, just diffuse through from high concentration to low concentration. Now, there are plenty of different substances that require the cell to open up channels in order to get through, and that's what we call active transport. In active transport, the cells must use energy in order to move the molecules. And it's kind of cool here, if the cells are using energy and they're putting forth effort, they can move molecules against the concentration gradients. So diffusion always moved with the concentration gradients from high concentration to low concentration. But if a cell is doing active transport, it can actually move solutes or substances from low concentration to high concentration. So it can move it against or opposite of the concentration gradient. So it's kind of opposite of diffusion and osmosis. So this active transport is taken care of by those transport proteins that we talked about in the cell membrane that are kind of fluid moving around within the cell membrane. They are the ones that are going to move those molecules against their concentration gradient. And one of the key kind of examples is the sodium-potassium pump that happens within or in the plasma membrane. It's required, kind of for the cell to keep a sodium concentration high on the outside of the cell and a potassium ion concentration high on the inside of the cell. And if diffusion were to take place, then the sodium diffuses into the cell because it's going, from high concentration to low concentration. And potassium would naturally diffuse outside of the cell. So we need these the transport proteins in order to keep pumping sodium out of the cell and potassium into the cell so that it keeps that healthy concentration gradient that's required for cell function. All right, sometimes cells just want to take in a whole lot of stuff at the same time. So that's what we call bulk transport. It is definitely an active process, meaning that it requires energy to do, because the cells are either going to spit out a whole bunch of stuff at the same time, or it's going to take a whole bunch of stuff in at the same time. So there's two main types, like I alluded to. Exocytosis, in which substances are moved out of the cell, and endocytosis, where those substances are moved into the cell. So let's take a look at some types of bulk transfer. So the first type we're going to talk about is phagocytosis. This basically means the cell is eating. Okay, so the cell engulfs particles and packages it in those vacuoles. So it kind of just takes it, surrounds the membrane around it, and then that membrane kind of pinches off and becomes a food vacuole inside the cell. So that's what we call phagocytosis. The picture on the right is a picture of an amoeba cell, and basically it took one of its pseudopod, which are kind of like a little arm, and it just like literally scooped up a bunch of food and just kind of engulfed it into itself. It's kind of weird, but that's what we call phagocytosis. So another type of endocytosis, bulk transfer, bringing stuff into the cell, is what we call pinocytosis, and it's kind of like phagocytosis, but on a smaller scale. The cells are drinking in a sense. They're not, eating lots of stuff. They're drinking small drops of liquids and substances. It's not, very specific like transport proteins at all. So it just kind of, engulfs a little bit of whatever's on the outside and brings it into, a vacuole. So like we see here in the pictures, in this endocytosis, the membrane, starts to pinch off. Okay, it basically pinches off and turns it into a vacuole. So all the stuff from the outside is now in, protected and inside that vacuole there. Some endocytosis is kind of just random. The cell will just kind of, I want you, and engulf, whatever's on the outside. The third type we're going to talk about, though, is what's called receptor-mediated endocytosis. So this is kind of, very specific endocytosis, where you've got a molecule that's going to bind to a receptor on the cell surface, and that's going to signal kind of like the okay to take in and engulf this particular substance. So again, some of it's just random, it'll just engulf whatever, and some of it is very specific, and the specific ones we call receptor-mediated endocytosis. So it's what happens with cholesterol. So cholesterol needs to get inside the cell. It's got little receptors on it, and it's going to come and dock itself with the receptor proteins inside the plasma membrane, and that's going to signal the plasma membrane to engulf it and take it inside the cell. So that is actually it for our discussion on the plasma membrane. We talked about the structure and all kinds of functions of the proteins and how it deals with osmosis and diffusion, active transport, and bulk transport. So what we'll talk about next is kind of how the cell works, how it gets energy, and kind of, how it does the things, how it gets energy for the things that it has to do. How does it get energy, for doing active transport and stuff like that.